Good morning, everyone, and welcome to the chapter on genomic library or gene library. And if you remember correctly, we have already discussed about gene library some time ago. That we were discussing about EMBL3A, and we would now try to read about genomic library and cDNA library together. Let's see if we can recollect what we have already read. But before going ahead, we will have a small introduction to it. We are learning about construction of recombinant DNA. Somewhere um, it would fall in this area, right? Prior to that, we have to get the gene of interest. We should have known why we want it, what we want it. This is actually our objective. And we should have, based on the scheme of how these steps, the remaining steps are going to be, we should have made a choice of the vector. And it also depends on the resources that we have. It also depends on many other factors, like if whether enzymes are there or not, technology is available or not, and so on. And then in this place somewhere, we are actually making the rDNA, right? We will go through that again so that it's a kind of summary of what we have been learning, and it also sets stage for genomic library and cDNA library. And here is about host cell. What kind of host did we choose? Why did we choose that? And if that is the host, how is it going to impose restrictions on the type of vector we have? And the host itself can be influenced by the type of gene, the source of the gene, and our objective of whether it is to just clone or to express. And many things will determine what kind of host we are choosing. Or they're all, it's like a plan all of a sudden comes about, because there is not one factor that is determining every other steps. And eventually we have put it into the host, and then we should be able to select or screen for the cells that we are interested in, the cells which have the recombinant DNA. We want to select these transformants from non-transformants, and we should be able to screen the recombinants from non-recombinants. I hope you're getting used to the terminology, and that should be fine. But let's just have a way of summary of the whole, how the genetic engineering happens, and different paths that are possible for it. First thing is gene of interest. How do we get the gene of interest? We might have seen a phenotype somewhere, and because of that phenotype, we would have decided, okay, maybe we need to find a gene that is actually causing the phenotype, and that is how we will have our own interest. So to get that, we can do it by restriction endonuclease digestion. Maybe if we are lucky, and if there are some unique restriction sites that would not cut the gene of interest, and then probably we will be able to clone it. But it's not always. It is a matter of luck, a chance that it might happen, and it's still a little difficult. How else can you make is another way of mechanical shearing. You can just take DNA and then try to digest it or degrade it by passing through little bit of pressure is one way of doing it, so that there will be shearing. Sometimes they also use, I think so, ultrasound or sonicators to fragment the DNA, and it is random cutting. And when we do that, the shearing, the type of ends that will come will always, we never know if it is blunt ends or if it is one side overhangs, and we can never be sure about what kind of overhangs are these and the sequence here. If we are cutting with a restriction endonuclease, then we are actually sure. So if we are using mechanical shearing, then it will cause the ends are, we never know if they are compatible or what it is. It's a lot of we don't have a way of trying to know, or so that predict what is it, and then it will cause problems for us to ligate into a vector. So in that case, probably you can use the polishing. How do we do polishing? We can use exonucleases to remove the 3' overhangs, or we can use clinofragment to fill in the gaps where there are 5' overhangs, and probably we can then get blunt ends, which can then be cloned. And we are going to discuss about how to get cDNA of mRNA, and that is another way of doing it. We will discuss about it, but we also had a small introduction here and there about what is a cDNA. There is another way. If the gene is not very big, I mean, if it is say couple of hundred bases, we can still try to synthesize the using direct chemical synthesis. We don't have to go through all these procedures. If we know the sequence, if the sequence is known, then we can go ahead with those things. But otherwise, it's not possible. And if we, as I said, if we know the sequence of it, then we could also choose to amplify those regions and then perform PCR so that we can then isolate it and clone. So the way how we are constructing the recombinant DNA, there are homopolymer tailing. If anybody can recollect the enzyme that is used, you can tell me so that it will be easy for everybody. Yes, homopolymer tailing. What is the name of the enzyme? Anybody? What is TT? Okay. Terminal transferase. It is an enzyme which will add multiple bases to the ends, 3' ends of the DNA, and whatever nucleotide you're giving, it will add. It is template independent DNA polymerase. That is the enzyme. So we can use for vector, we could have used GTP as substrate. For fragments, we could have used CTP. So because they are complementary, we might be able to get a recombinant DNA. The ligation by cohesive termini is having identical sites. See the overhangs when they are identical, which can be generated by cDNA synthesis, or we can also do it by restriction endonucleases. We can also try to do by using linkers and other things, cDNA as well. We can there use simple ligation reaction because the overhangs are compatible. Then we can perform that. Blunt end ligation can also be done where we can make blunt ends and then try to use those things. Here are linker molecules. We can also, what is not mentioned is the adapters. They can also be used, and there is TA cloning as well. TA cloning, Taq polymerase, when it makes PCR, it adds a single 3' adenine like this. So if we could generate a vector which has single T, then these two can be ligated and recombinant DNA can be made. So this is a kind of summary of many things, that tools that are available to us, or the strategies that are available to us. And then we have a choice of the host. When we were discussing about vectors, we did discuss about many of hosts. E. coli is the most common one used, majority of the plasmids, but there are plasmids are manipulated. Majority of the vectors are manipulated in E. coli. But there are several others, including yeast. There are also Bacillus subtilis, and some of the enzymes, say E. coli is gram negative and others may be gram positive. So if you want something, a vector for gram-positive one, maybe you would have to choose some other DNA host. So animal cells can also be used, and so on. So how do we put DNA into the host is the question. There are, this is a word here, it is transfection, and there is another one that is transformation here, that is another word. Transfection is when we are using phage to put in the DNA into the cell, then we are calling it as transfection. Transformation is, so transformation is direct DNA is given in here, then you would say it as transformation. And the other one is, of course, in vitro packaging and into the phage, and we'll say it as transduction. Okay. Yes, I hope you have a good enough idea about these. And we now need to also figure out which ones are the, so let me just say we have made the rDNA, and when we are making the rDNA, we would have performed ligation. So in that case, in the rDNA here, we would have had the gene of interest cloned in the ligation. But some of them would be without any fragment. They would have just been empty vectors, vector-vector religation. And when after this, we would have had a step that is transformation. When we are performing transformation, we would have taken all this, both the mix together, because they are all happening in one single reaction. Some of them are recombinant, some of them are non-recombinant. And we would put it into host cells, say, for example, E. coli transformation, simple. So some of the E. coli would get, there will be at least 3 types. So some E. coli would get the DNA, and the DNA could be either recombinant or non-recombinant, right? I hope you remember when we discussed about blue-white screening or PBR 322. I think we should we did discuss that also. So these two would be called as transformants. That means they have acquired DNA. And this majority of the cells would not have received DNA. Those would be called as non-transformants. So within the transformants, again, we have 2 categories. This is a recombinant, right? This is what it has acquired. The other cell is a non-recombinant. What we need out of all these is that we are specifically interested in this particular, these cells, right? This is the cell that we want. And majority of them are going to be the non-transformants, which did not get any DNA at all, and we don't want them, and somehow we should get rid of it. And when we were discussing about vectors, we did discuss that we need to have selectable markers, right? We need to have selectable markers. That is usually antibiotic resistance genes. They will eliminate all the non-transformants. They will eliminate them. How do they do it? We would have taken a plate with nutrient agar and add, say, for example, ampicillin in the plate. And we would have had a marker on the vector, which would have been ampicillin resistance gene or beta lactamase. So when these cells are plated, and some of them will form colonies, and those that have formed colonies will be the ones that have the DNA, the vector. Either it could be recombinant or non-recombinant. So if they have the vector, they must have had the ampicillin resistance gene. If they have the ampicillin resistance gene, they would have grown here, otherwise not. So we will be able to eliminate all the non-transformants. And now our question is to identify recombinants from that of non-recombinants. And for that we can have, so before going there, this is selection. We are selecting transformants from non-transformants. That means selecting means only when it says selection and screening, there are two terms used sometimes interchangeably, but I think the essence of selection and screening is, when you say selection, like in natural selection, only those with an advantage can grow. It is something like the survival of fittest. The rest will die out. They will not grow on the plates. Screening is something like both. I'm just giving an example here. Assume that both transformants and non-transformants both will grow. But by looking at them by visual observation or by some other additional tests, we can figure out which of them is a transformant and which one is not a transformant. Okay, we can figure out those things. Say, for example, we will take blue-white screening. Typically, in a blue-white screening, what do we have? We have lag Z prime, truncated one, and ampicillin resistance marker, resistance gene, or I will just put it as AMP resistance gene. And we would have cloned here, and I hope you can recollect about alpha complementation. And the other term or the phrase is insertional inactivation. Right? So their ampicillin resistance gene is for selection. That means when you plate these cells, either recombinants or non-recombinants, upon ligation and then transformation, you plate it on LA or nutrient agar with ampicillin. Those that have acquired the vector, either recombinant or non-recombinant, will grow. The rest will perish. And that is selection. And if you have done this on XGAL, and you can figure out what is a recombinant from non-recombinant visually. But by looking at the color of the colonies, right? So if it is a recombinant, it would look white. The colonies would look white. And if it is a non-recombinant, it will produce a functional alpha component, because of which beta-galactosidase is arranged or formed, and then it would hydrolyze or act upon XGAL to give rise to blue color. So here, by looking at the color of the colonies, either it's blue or white, we are able to figure out if it is a recombinant or non-recombinant. And that is screening. Okay. So we got all the cells, and from them we are able to figure out which one is a recombinant and which one is not a recombinant. That is the difference between selection and screening. In screening, only those that we actually want will survive and the rest will die. In screening, all will grow, but we are able to figure out which ones are the ones that we are looking for, the recombinants usually. Okay, so I hope you got the terminology right. And coming back to this slide where we are trying to summarize different strategies. We already have good introduction to majority some of these selection and screening strategies. And before going ahead, it's good to revisit them. And for selection and screening, we use several techniques, and one of it is hybridization. It's usually what it means, hybridization, when we are in the context is about the hydrogen bond formation between complementary sequences. In this case, if you look at northern blotting or southern blotting, we have used hybridization. The probes are nucleic acid, and we are trying to figure out nucleic acids, and that is by hybridization. We can also figure out this is hybridization is a screening method here, right? It's a screening method. PCR is another way you can figure out. We will discuss about it maybe in the following slides in today's class or tomorrow, where, say, for example, you can do colony PCR. An example of it is colony PCR. And we can find it out. Remind me if I didn't touch upon it, but otherwise we will discuss it in this as a part of this lesson. Immunochemical is using antibodies, monoclonal antibodies, and antibodies raised against a particular protein of our interest. So if the gene is present, protein is produced, protein could be produced, and that presence or absence of that protein can be done through using antibodies conjugated to proteins and so on. The same way, you can also have protein-protein interactions and protein-ligand interactions. Functional complementation is like the blue-white screening we have just discussed. And gain of function is also, simply to put it, I'll just erase this, and simply to put the gain of function is something like this. If you remember some of the examples like marker genes, we discussed about TRIP minus, I think, right? That means a cell is there. You have taken, say, for example, yeast. And yeast, this one cannot is TRIP minus. It is oxotropic to tryptophan, so it cannot synthesize tryptophan. And if you are growing it in minimal media, it will not grow, no growth. Why is that? It is unable to supply, it's unable to make tryptophan. And if there is a way to grow, we should have put, if we put it in minimal media, you should have supplemented the minimal media with tryptophan, and then it will grow, right? So what do we do? How do we employ this oxotrop as a, and which kind of selectable marker can we use? We could have taken a vector which has a functional TRIP gene. That means it has the ability to, the missing link or missing enzyme to produce tryptophan. And we would then try to transform into the host TRIP minus. And now we are going to plate it only on minimal media. And if you are plating it on minimal media, those cells which have gotten the DNA, the vector, would grow because it is, the vector has the gene for tryptophan production. So even if you put it on minimal media, that cell can, those cells can grow and form a colony. Whereas the other ones, because they do not have the capacity to grow on tryptophan, on minimal media like this, they will not grow. So that is functional complementation, or you can, that is gain of function. Similarly, you can also take it as, take, for example, antibiotic resistance, what we are discussing about the AMP here, ampicillin resistance. That can also be used as an example for gain of function. Okay, so why we are discussing all these things is, um, if we did not have any sequences, if we did not have any sequence, excuse me, sorry, say we don't have any sequence information, all we know is a phenotype and no sequence information. How can we get the gene of it, right? In that case, we don't know the, say for example, we don't know the sequence, we don't know location where it is, and we should be able to figure out the gene of interest. How can we do that is the question. In those cases, we can take approaches that are something like the genomic library. If I remember correctly, we have discussed about two different strategies, right? One of it is you have a tree and there is a branch. On that branch, you have a bird, and you would take a gun, or probably if you want to get it, okay, or something, and try to shoot it. And that is how you get it. And in some cases, all you know is there are some chirps or noises coming from bush, and you don't know what is making the noise, and you want to get something out of it. So in that case, what you do is put in a stick and see. You will, that is what you call it as the beating the bushes, and see whatever comes about, and maybe it is a bird that is of interest, and then you catch all those that come out, right? Here in the first one, where you target something and shoot something, you have information of it. The some properties of it is there. You know the location, you know the shape, you know the size, and some other things like that. Then this is the direct shooting kind of approach. In other cases, you don't know what is there, but you are supposed to figure out. And then what do you do is a general strategy where we just try to beat the bushes and get some things, whatever is coming out, we will then take it and use it. So genomic library and cDNA library are something like that. What you do is you take, this is the genome, and it has, say for example, 100 genes. You take all the 100 genes, you clone all the 100 genes, all the 100 of them. And that is what you call it as the gene library or genomic DNA library. And then you test each one, or by using other strategies, you test whether this particular one can give the phenotype that we are looking for or not. And that is how this is the screening part. Right? I hope you understood the general scheme of it. Because we don't know the sequence, because we don't have enough information about it, we will clone all of it, clone all the genes, and then we should be able to screen the library for gene of interest or phenotype of interest. So there are multiple steps in it. We don't have to really go through it. We will, as an example, we already discussed TMBL3A. In that, we will revisit that. But otherwise, simply is, as I said, as I gave it here, first we will isolate the DNA, fragment it, and we will construct the clone using a suitable vector. And then we will plate, and then we will screen them all. Okay, this has some advantages. Say for example, if I made a gene library once because I want to find gene X, and I made a gene library and I found out a clone with gene X, then my objective is fulfilled. In the future, I might want for gene Y. Then I can use this same library again to get clone with gene Y, right? You can do these things multiple times and see, use the library for many different times or purposes in the future as well. So genomic library construction is, or was, and still is, a good strategy in many labs. Okay. So I will quickly go through this, and I hope you can recollect it. This is about EMBL3A, and this is the genomic DNA. Some of the differences that we have discussed, I think that is the best way to look at, because we already discussed thrice. Can anybody discuss why we are using phosphatase to treat the genomic library? Can anybody explain that? So we have taken genomic DNA and we are digesting it with South3A, and it is partial digestion and phosphatase, and then it is treated with phosphatase. Why are we treating it with phosphatase? Can anybody try to explain? Yes? Okay, in the last class, in some class, we have discussed one of the objectives of genomic library is that we want to know about the whole genome, right? We want to know about the whole genome, and at different places, we may have A, B, C, D, E. We want to know the how many genes are there, or all the genes. Assume we will, how many genes are there? And the second one would be, what is the order of the genes? For us, we usually just simply take A, B, C, D, like a series, but we don't, with the gene names are often different names. And we should try to know the order of the genes. So when we make, we make partial digest and we don't use phosphatase, at one point we are going to add ligase. We are going to use ligase here. And when we use ligase, there is always a probability, or I will just take a step back. Say, for example, we have digested it, South3A, so it is cut into several of these pieces like this, and now we have several clones. And if we clone, if the next step would be after digestion, we are going to add ligase. If we added ligase along with the vector, okay, assume this is vector. We discussed about the way we talk, right? Left arm, spacer, right arm, L, S, R, left arm, fragment, and right arm, and so on. In that possibility, there is also one of the possibilities, the ligation of A and B, A with A with C, A with D, A with E. Like that, we can actually have many other combinations of products that could form. We don't want that because it will compromise the which order of the genes, what is the order of the genes that are present in the DNA, in the genome. We would never know that, right? So we don't want these kinds of fragments to form because it will complicate our whole study. So therefore, we will treat it with phosphatase so that we will eliminate the formation of all these fragment ligations. And that is one of the reasons why we treat the fragments with phosphatase. Otherwise, in majority of the cloning procedures that we have learned, we had learned that it's always because that the vector is treated with phosphatase because we don't want vector religation which may show up in our selections. And it will complicate our screening procedures as well. Okay, that is about this step. Otherwise, we have EMBL3A, SAL1, BAMH1, ECOR1. Similarly, we have the other ones. I hope you remember about red and gam. Red and gam are for SPI phenotype. SPI is sensitive to P2 inhibition, right? That means lambda phage cannot infect a lysogen that is already having a P2 prophage. That is, P2 somehow inhibits the lambda phage infection on P2 lysogen. Okay, that is SPI phenotype. You can go back and look at the older videos, or you can always ask specific questions if you have any. And then we are treating with BAMH180CoR1 because we want to get rid of these polylinkers, and that is done by isopropanol precipitation because they are very small. The larger ones will precipitate faster, whereas the smaller ones will precipitate slower. And then we will ligate, and we know when we put it on P2 lysogen, only those that are recombinant, these will grow or form plaques, and those with the stuffer region will not form plaques. First of all, these will not form. They will not form in higher proportion because the polylinkers are removed. Even if they formed, they will not form a plaque because the red and gam that are present on the stuffer region would not have allowed the formation of the plaques. Okay, so that is the general how a genomic library is constructed. You take the whole DNA and you collect all of them, and eventually what you will have is a whole plate of plaques. And then we would have taken the plaques and screened for whichever properties they have, and then we can figure out the gene of interest, or we can probably figure out the order of the genes as well. Say if each gene A is in one clone, B is in one, C is in one, how can we tell the order? We cannot actually tell the order of it. Let me see if we have another one. I will come to that. But before, maybe at least I will try to introduce you this. It says partial digestion. Why partial? That means you are actually taking the whole genome and you're taking many copies of them, right? So assume each one is a copy. And we are saying partial digestion with the Sow3A. That means Sow3A is a 4-base cutter, which will cut quite often. And we are making it only partial. That means in one gene, it may cut, in one genome, it may cut something like this. In another one, it may cut something like this. In different of these, you will have several cuts in different positions, right? This is how the cut may be made. Because it is Sow3A, frequency is high, because it's a 4-base cutter, but you're allowing partial digestion only. So in one case, you may have a fragment that is covering, say, for example, gene A, gene B, gene C, gene D, gene E, gene F, and gene. So if you see like this, A and B can be cloned together in some cases. In some cases, you may have B and C cloned together. In some cases, you may have C and D cloned together. And that way you can figure out what are the genes that are cloned together. And when you analyze it, you can figure out the order of the genes. We have a technique called as chromosome walking. We will study about it. I will try to make up a problem for it, and then we will try to solve it that way. Theoretical problem, but it should help us to understand the principle of the technique. Okay, in the next class, we will discuss about another one, a lambda Charon, using a vector there. It's a replacement vector. I hope you remember what is a replacement vector and what is an insertional vector. I'll stop here, and what I will encourage you all is to please come up with some questions that you may have for future. So now you are in fourth year, and you will be moving ahead. And when we are in transition, say from plus two to what to do next, we have these transitions. That is where our stress levels usually get very high. And it gets even higher. It is at the end of BTech, because we don't know the paths. The paths are too many. Like, should I take up the job? Should I go abroad? Should I stay here and do MTech? Or what to do is always a problem. The transition is, this is a highly stressful one, because we don't know if we are doing it right or wrong. And that is why I would like to one day discuss with you. And as I said, I'm not going to provide solutions. All I can probably do is provide ways of thinking for it and different things you need to consider, so that you can make best decisions in the situations you are in. Each of us, our lives as such, we have different scenarios, different contexts, and it is unique. And who is the best judge is usually the person who is experiencing it. But the one experiencing it doesn't know, not necessarily. I didn't know many things when I had to make these decisions, and good or bad is different. Usually it is like this, as I would repeat also in the next class whenever I discuss about these career opportunities, is there are no right decisions. There are only decisions that can be made right. Okay, there are no plans. It's something like you have to have a path and work until it gets right, or work until you win. That's the only way you will succeed. So why I say that is, you can plan as much and make a fantastic plan up till getting a Nobel Prize. But if you didn't work on it, it doesn't. It's useless. You can make a very bad decision, or say a plan how to open a restaurant. Studying biotechnology and open a restaurant, it's kind of bad decision in my view also, but not necessarily. But when you work hard towards it and your restaurant becomes the best in the town, then that is the best decision. So what works is, you decide, and the input that you put in, that is going to define what is happening. There are many other factors like luck and so on, which is beyond our control, and we cannot do anything about it. Okay. Well, please feel free to ask any questions, and then we can have one class at least to discuss on that. And so hopefully it will be helpful to you and me as well. Thank you. Take care and have lots of fun. Any questions, by the way?